The overall goal of this procedure is to attain an exhaustive characterization of a gene family in plants through a global approach, including the definition of gene structure, conserved protein motifs, chromosomal location, gene duplications, and expression patterns.
This method can help answer key questions in the classification of plant gene families, such as whole genome identification of family members and their transcriptomic profiles through gene expression meta-analysis. The main advantage of the whole procedure is to provide an exhaustive list of experimental steps to achieve a complete gene family characterization.
This procedure can be applied for any gene family of any plant species for which genomic data are available. Moreover, this global approach provides valuable information to identify interesting candidates for functional studies.
Demonstrating the whole procedure will be Pietro Ariani from my laboratory.
Open the BLAST web page and click on the Protein BLAST section. In the Enter Query Sequence field, enter the amino acid sequence of the protein that will be used as the probe to identify the other family members. Use a protein that has all the important features that characterize the family. Next, in the field Choose Search Set, select the reference protein database and the organism of interest. Now, in the field Program Selection, select the PSI BLAST algorithm. If desired, click on Algorithm Parameters to adjust advanced parameters such as max target sequence and scoring matrix. The E value is the threshold, which for this search is decreased from 0.005 to 0.001. Click the BLAST button to run the analysis.